Hello, anatomy students. Now that you've learned how an action potential is generated down an axon, we need to learn what happens once that action potential actually reaches the axon terminal and how that signal is transferred to a neighboring neuron. So there is a note sheet that goes along with this. So grab a pencil, grab that sheet. If you need to put this on pause, then go ahead so you can get all that stuff. All right, the first thing that I want to talk about is that we have electrical synapses between cells in our bodies, and we have chemical synapses between cells in our bodies. With an electrical synapse, we usually have particles with different charges that are going to move through gap junctions from one cell to another. But in the nervous system, we're talking mostly about chemical synapses. So this is where our presynaptic cell is going to send a signal across a gap that is received by our postsynaptic cell. And in this case, the signal is a neurotransmitter. So go ahead and fill out these squares as I'm jib-jabbing at you on this video. So once an action potential arrives at the axon terminal, it's going to stimulate the opening of a voltage-gated channel, a calcium voltage-gated channel, that's going to allow calcium to enter into the cell. And what our calcium does is it's going to signal these molecular motors. So you can see that on the presynaptic cell, like over there by the number 2, where you have like those red lines. These molecular motors, I like to think of them as like chairlifts at a ski resort, where they have proteins instead of chairs that actually bind to vesicles and carry them from our cell body where the neurotransmitters and the vesicles are created, and then they carry them all the way down the axon when they're stimulated to, in order to allow for those vesicles to actually reach the inside of the cell membrane very quickly. And then via the process of exocytosis, these bubbles, these vesicles, are going to release their neurotransmitters into the gap between our pre and postsynaptic cells. That gap is called the synaptic cleft. Then at number 3 in our picture, our neurotransmitter, which is our signal that has just left the vesicle, is now going to diffuse through the cleft and bind to a receptor on the postsynaptic cell. And in doing so, it's going to initiate a local potential. And so a couple of things that I just wanted to review on that with you is that the neurotransmitter is acting as a ligand, and the channel that it binds to is a ligand-gated channel. So it wouldn't open unless a neurotransmitter actually bonded to it. And then when it opens, it's going to allow sodium to enter into the cell. And once that happens, the sodium coming in, remember that's positive, the inside of the cell is negative, and the sodium coming in is making that general area more positive. That's going to cause a local potential to move from the dendrites on our postsynaptic neuron through its cell body and then all the way down to the axon hillock of that of the next cell. And remember that at the axon hillock, an action potential will be generated if threshold is reached. And remember, that threshold is negative 55 millivolts. So after the neurotransmitters bind to our postsynaptic cell, they can't just stay there. What has to happen is they need to move. Otherwise, we are constantly bothering that postsynaptic cell. We're stimulating it over and over and over and over again, and that might not be something that we want to do. And so there are a couple of things that can happen to our neurotransmitters. They can simply diffuse away. They can be degraded by an enzyme, or they can be taken back in and recycled by our presynaptic cell. When we're talking about neurotransmitters, one of the ones that we talk about a lot in this class is going to be acetylcholine. And acetylcholine, after it binds to our the ligand-gated channels on the postsynaptic cell, it gets broken down by an enzyme called acetylcholine esterase. And part of that molecule is recycled by the presynaptic neuron, but a part of it can like diffuse away and go and be used by other cells, maybe not even other neurons, just other cells in our body. And that's everything that I just said. Okay, so the next thing that I want to talk about are these different kinds of postsynaptic potentials. I mentioned this a little bit in one of the earlier videos, that some postsynaptic potentials are excitatory. The ones that allow for I'm going to move myself out of the way here. The ones that allow for sodium to enter into the postsynaptic cells are called excitatory postsynaptic neurons, or EPSPs. These excitatory neurotransmitters are going to bind. They cause ligand-gated channels to allow sodium in, and there's a temporary depolarization of the area, and it's going to cause the surrounding areas to depolarize. So basically, it's going to cause an excitatory local potential to kind of move from one place to another until it goes to the axon hillock. But some neurotransmitters don't cause EPSPs. They cause IPSPs. IPSPs are inhibitory postsynaptic potentials. When an inhibitory neurotransmitter binds to a postsynaptic cell receptor, it's going to cause ligand-gated potassium channels to open. But now potassium doesn't go into the cell. Potassium moves out of the cell. So now you have positively charged things leaving the interior of the cell, moving to the outside. And that's going to cause the inside of the cell to become more negative. It's going to cause a temporary hyperpolarization or an over-separation of charge. It's going to cause, like, the graph on the voltmeter to kind of dip downward instead of making a hill moving upward. And it's going to cause the surrounding areas to hyperpolarize. And those potentials also are going to move, along with any EPSPs that are occurring, down to the axon hillock. So there at the axon hillock, we are going to have a summation of all EPSPs and IPSPs that are being produced at the same time. So they all add up at the axon hillock. They will produce an action potential if the magnitude is great enough, if our threshold is reached. And this is just going to allow us to not have to respond to absolutely everything. This is how information can be processed as it moves through the neurons. And remember, like, not all sensory information that we get is noticed by us. It's going to be kind of some of it is going to be put on the back burner. Some of it we're going to ignore. But these are things that we don't have to consciously do. Our cells are doing that for us with this whole idea of threshold, and is a stimulus strong enough to create an action potential to actually say, hey, this is important, you've got to respond to this. So this is the way that our bodies can kind of control what we respond to and what we don't. So remember that the summation of EPSPs and IPSPs are kind of like adding together positive and negative integers. And so if you have more EPSPs than IPSPs, or if the EPSPs have a greater membrane potential, a greater change in the membrane potential than the IPSPs, and threshold is reached, then we're going to get an action potential. But if the opposite is true and the axon hillock doesn't release action potential, and doesn't release threshold, then no action potential is generated. Remember, you can't get like a little tiny action potential. Action potentials are all or nothing. All of them are going to reach a height of like positive 30-ish. None of them are just going to be like negative or positive 25. No, they're going to go to the same height and come down to the same value for hyperpolarization. They're not going to vary in that way. The action potentials are all or nothing. And in order to get an action potential, threshold must be reached at the axon hillock. And so here, this is just a little example of what can be happening. So at negative 70, we have our resting membrane potential, and now we get an EPSP, which is shown in green. And then we get another one, and together they're kind of raising our our membrane potential to become a little bit more positive, but has it reached threshold yet? No, not yet. And look we get an IPSP at the same time, and that's the pink little dip in the graph. And then, we get another EPSP and then another IPSP, but then we get an EPSP that is a little bit greater in magnitude, and that's going to push our axon hillock up to threshold and allowing the rest of that yellow line, the action potential, to occur. Oh, wait, I'm going to go back to that so that I want to make sure that you guys can see the caption at the bottom. So if you need to pause this to read it, then go ahead and do that. Okay, so we have two different kinds of summation to know. The first kind is spatial summation. With spatial summation, we have multiple PSPs, or multiple postsynaptic potentials, at the same time, but they arrive in different places on the axon. Regardless, they all travel to the axon, they add up, and if threshold is released, bam, we get an action potential. But spatial summation is defined here at the bottom of the slide. This is an example of spatial summation. So you get simultaneous stimulation, maybe by several presynaptic neurons, all at the same time. They're just coming in at different places. If they all add up at the axon hillock and threshold is reached, then number 3 happens. If they don't reach threshold here at the axon hillock, then number 3 does not happen. Our final kind of summation is called temporal summation, and this is when multiple PSPs, or postsynaptic potentials, are arriving in rapid succession. So they're arriving at kind of different times. We're talking about milliseconds here, folks. It's not 5 minutes in between. But with temporal, we've got maybe one presynaptic neuron that is stimulating in quick succession our postsynaptic neuron. And all of those move to the axon hillock, they add up, and again, if they reach threshold, then we get an action potential. If they don't, then we won't. So with temporal summation, we have just one axon repeatedly stimulating our postsynaptic neuron, one right after the other. And this is what happens with temporal summation. So we just have one axon, one that is stimulating our neuron at a high frequency. And again, if all of those add up to reach threshold at the axon hillock, we get an action potential. And if they don't, then no action potential for us. Okay, so that's the end of this lecture. I hope you enjoyed it, and I hope that you learned a lot, and I will see you again soon. Take care.